Okay, well, welcome everyone. I'd like to begin today by acknowledging the traditional owners of the land on which we meet, and I'd also like to pay my respect to elders past, present and emerging. So welcome everyone. For those who don't know me, my name is Beck Sheehan. I'm the Director of Cure Research and Programs at FightMND, and I'd like to thank you all for logging in to the FightMND Scientific Seminar for October. We have a really exciting presentation lined up for you today, and it'll highlight some of the translational research capacity in the MND space. So following on from the presentation today, we are leaving plenty of time for discussion and questions. So if anyone would like to ask anything, please use the reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know when to ask your question. Alternatively, you can use the chat, where our team will be monitoring and read out the questions on your behalf. And please note that the chat function is only enabled if you're using the app-based version of Teams, and unfortunately does not work in the web-based version. So if you're using the web-based version, you'll need to raise your hand to ask your question. So today I'd like to introduce our speaker for today, Associate Professor Brad Turner. So Brad is the head of the Neurodegeneration Movement Theme and MND Laboratory at the Florey Institute of Neuroscience and Mental Health at the University of Melbourne. Brad obtained his PhD in biochemistry and molecular biology at the University of Melbourne, followed by a postdoctoral training at the University of Oxford under an NHMRC CJ Martin Fellowship. Following this, he was recruited back to the Florey to establish his independent research group, supported by a Stafford Fox Foundation and an NHMRC Dementia Research Leadership Fellowship. He leads a large and ambitious research program with many projects and initiatives, and amongst these is the MND drug screening platform, which he's going to tell us about today. So thank you for taking the time to speak to us, Brad, and over to you.
Thank you, Beck, for the kind introduction and invitation to speak today. So today I'm going to talk about one of the research programs that my lab is running, and it focuses on the use of patient stem cells as a drug screening platform for MND. And let me just advance my slides. So I just thought I'd put just 3 introductory slides just to put this program into context. So throughout this talk, I'll be referring to MND as amyotrophic lateral sclerosis, ALS. And this is a disease which affects around about 2,000 to 2,500 Australians. Here are some famous faces of people who have lived or living with MND, including Neil Danaher, Pro Hart, the less known one, and also Peter Doan. This is a late onset disease, and it's caused by the selective death of motor neurons in the brain and spinal cord, and it's typically fatal within 2 to 5 years, and no effective treatments or cure. I really wanted to emphasise that this disease is that the cause of this disease is largely unknown. So if we look here at this pie chart, most of the research world has been focused on this slice of the pie, and this is familial inherited MND, of which we now associate about 50 genes with familial MND, and it's really this part of the pie that's been largely neglected through research, the sporadic MND. The other point I'll make is that in Australia, at least, there's only one approved drug for ALS, and that's Riluzole. And Riluzole has been around, was FDA approved back in '95. And this is a drug which dampens a process called excitotoxicity. So it's thought to act at the synapse, reducing this destructive glutamate-mediated excitotoxicity. Okay, so that sets the scene for this question. So there's been about 100 or so randomized controlled trials using proposed disease-modifying drugs in ALS, and all of them have failed to show positive results in the past 50 years. So this is quite a sobering observation, and the question then is why? Why is this disease so notoriously difficult to treat? And I'd like to talk about 5 different points that I think are relevant to this. And the first is we all know that no 2 cases of MND are identical, and so we often talk about this clinical heterogeneity of ALS, in that it has many clinical presentations, as shown here. We have limb onset, bulbar onset, and then we have variants, PMA, PLS. We have symmetrical, asymmetrical presentation. We have the flail arm, flail leg variant, and all of these have different clinical trajectories in terms of prognosis and symptom presentation. Another way that we can depict this is using this type of plot here. This is a spaghetti plot. And what we're looking at here is the relation between time from clinical visit, and this is the ALS-FRS score. And you can see that these patients all have unique disease trajectories. There are patients that will succumb to their disease within the first year and others that will live out for 7 years. So the message here is enormous variability in clinical presentation. Now, underlying all of this clinical heterogeneity is pathological heterogeneity. And we know that TDP-43 proteinopathy is a major hallmark of this disease. But even within TDP-43 proteinopathy, we have diversity. So if we look here at TDP-43 aggregates, we see different morphologies in different regions of the CNS, so filamentous, round, conglomerate. And there are forms of ALS that don't have any robust TDP-43 pathology. I've mentioned this before, so we see this vast genetic heterogeneity of ALS, particularly underlying familial ALS. And this graph just shows you that there's been a tsunami of genetic discoveries over the last 3 decades. And the major gene players are shown here in these circles. So SOD1, TDP-43, C9orf72, and FUS, and there are more to come. So this adds to the complexity of the ALS puzzle. And what are the genes encode? What does this lead to? Well, overall, we think there is vast biological complexity in ALS pathogenesis. And this is nicely illustrated here in this diagram, where we have a lower motor neuron, which is affected by these pathological processes, and that is then complicated by the action of glial cells such as astrocytes and microglia, and of course the upper motor neuron, which is barely pictured here, but it just is an equal or perhaps as an important player as the lower motor neuron. So as scientists, we love laboratory models, and this is a really nice depiction of the strengths and weaknesses of different lab models. So we know that live patients, living subjects or participants rather in these clinical trials have the most relevance to the human condition. So ALS is uniquely human. However, we do use mouse models primarily in ALS research. And although these models can give you more mechanistic insight than patients who are already diagnosed, they lack that human clinical relevance. If we look here on the cube, we can see the emergence of these technologies, such as induced pluripotent stem cells. So these have high human relevance, high mechanistic insight, and I would say medium, potentially high throughput. So for the purposes of today's seminar, I'll be talking about this area of the cube, which is induced pluripotent stem cells, and I'll also touch on some organoid work that my group is doing. So I guess the question is, can we use patient iPSCs as a tool for drug screening for motor neuron disease? And I'm just going to illustrate a bit of background on iPSC technology, give you some case studies on where it has been used successfully, and then show you our program, into iPSCs. So I would say that iPSC technology is relatively new, and it all started with Yamanaka and colleagues who published this paper in Cell, and they demonstrated that you could take somatic cells from a patient, and back then it was typically skin cells, but now you can take PBMCs, saliva, urine, there's many sources of somatic cells, and using a combination of factors known as the Yamanaka factors, you can reprogram these cells into iPSCs, and once you have them in this pluripotent state, you can then differentiate them into your cell type of interest. So for MND, we're interested in neural cells. And what was interesting was it took about a year for this technology to be embraced by the ALS research community. And there were studies led by Kevin Egan at Harvard, where he took, I think there were 2 Italian sisters with SOD1 mutations, and he published the first demonstration of iPSC technology in ALS. So he's able to take these skin cells, reprogram them into iPSCs, and then differentiate them into motor neurons. And these motor neurons looked morphologically correct. They had expressed the right markers, such as HP9 and cellular markers. So this was the first demonstration of the application of iPSC technology for ALS. The question then is, what disease phenotypes can one elicit from this model system. And it was interesting that it took about 6 years before the first phenotype was reported in these cells by the same group. And what they did here was they patched these motor neurons in the dish and were able to show that the excitability profiles were altered in the presence of a SOD1 mutation. And you can see that from this trace, we've got this increased train of action potentials indicative of a hyperexcitable state. So I'd point out here that this is quite a subtle phenotype. It wasn't as dramatic as one may have expected, but it certainly led and had very important implications for the field. And now I wanted to talk to you about 3 case studies where ALS patient iPSCs have been used to inform drug candidates that have gone into clinical trial. So the first case study is related to that paper I just mentioned. And what the authors did is they took a very well-known drug called Retigabine. And Retigabine is a calcium channel opener. It's used as an anti-epileptic drug. And they showed that application or treatment of these SOD1 iPSC-derived motor neurons with Retigabine was able to attenuate this hyperexcitable phenotype. And it does so, as I mentioned, by opening calcium channels in motor neurons and relieving that excitability. The second case study I'll show you is one involving a drug called Bosutinib. So Bosutinib is a tyrosine kinase inhibitor. It's used for the treatment of, I think, CML, various leukemias. And what this group did is they, again, took some SOD1 patient iPSCs, differentiated them into motor neurons, and they conducted a drug screen using about 1,400 FDA compounds. And it turns out that this drug, Bosutinib, stood out from the rest. And what they were able to show was a dose-dependent increase in survival of these motor neurons treated with this drug. And I mentioned that Bosutinib is known as a Abl kinase, and it's thought that it leads to neuroprotective pathways. The third case study, and this one's quite well known in the ALS field, and that deals with a drug known as Ropinirole. And this was a paper published 4 years ago. Again, this is an existing generic drug. It's a dopamine 2 receptor agonist, so it's used for Parkinson's. What this group did is they took their library of ALS iPSC lines, performed a drug screen. It was quite an in-depth study. They looked at multiple outcomes, and what they found was Ropinirole was superior, and it worked quite well in FUS and TDP43 ALS patient-derived motor neurons. Its action in ALS is thought to be different to Parkinson's disease, so it's thought that it's taken up by mitochondria where it exerts antioxidant effects. So these are 3 really exciting examples of how patient iPSC-derived neurons can inform drug candidates. And I just wanted to point out that each of these drugs has led to a corresponding phase 1 or phase 2 trial. The most notable one I would mention is Retigabine, also known as Ezogabine, where they showed target engagement. This drug was able to attenuate hyperexcitability of motor neurons in these ALS patients, as shown by Steve Vucic, Matthew Kinnan, and colleagues. So this is quite exciting. iPSCs, like any model, have limitations, so one needs to be aware of these. And I would say the first major limitation of using this technology to model ALS is the undeniable immature nature of these cells. So this was a great study performed by Klaus Fedson a few years ago when he took iPSCs from patients and he compared the transcriptome to motor neurons, which are laser captured, microdissected from adult spinal cord tissue. And what you can see from this heat map is that the iPSCs have an almost reciprocal gene expression pattern to mature motor neurons, which were isolated from human postmortem tissue. So it is undeniable in the field that these cells are immature and fetal and probably equivalent to second trimester motor neurons. The second obvious limitation of iPSC technology is these cells they're monocultures in 2D and they lack the complexity of the human CNS. And that's illustrated here. So this is a motor neuron. We know that ALS is a disease of neighbours, and we know that astrocytes, microglia, myelinating glial cells and interneurons all impact on this disease, and they are completely absent in this simplistic cell culture model system. But there are ways around this. The third point I wanted to mention was iPSCs are not perfect in predicting drug targets, and I'll give you an example here. So about 10 years ago, there was a paper published which suggested that a cell death pathway, an inflammatory cell death pathway called necroptosis, mediated motor neuron death in iPSC models of sporadic and familial ALS. And it was largely through the use of these inhibitors, necrostatin 1 and NSA, which block necroptosis. So my group and others have shown that if you perform a genetic experiment where you genetically ablate the critical mediators of necroptosis in an animal model, you get absolutely no benefit in terms of disease progression and motor neuron pathology. So I think this is an example where iPSCs were not that informative and have led to an irrelevant drug target, I think, for ALS. So my group decided we wanted to venture into using iPSCs a few years ago, and we launched an initiative, and the initiative is called ID MND, standing for Ian Davis MND Initiative. And this is the world's largest precision medicine program for MND. And it contains the largest library of MND patient stem cells in Australia for research purposes, and this includes disease modelling and drug screening. And it's certainly the largest effort in Australia to profile and understand MND at deep levels, so at genetic, molecular, protein, metabolic and clinical levels. And the idea behind this initiative was we wanted to fast-track drug screening for MND to accelerate the search for effective treatments for MND patients, so really speed up that clinical trial pipeline. So the idea was quite simple. We wanted to take this patient iPSC technology and combine it with what I would say is industry standard robotic screening, automated screening platforms. And this is just a video here showing you one of the robots we've got housed downstairs involved in this program. So we'll just we'll let that robot complete its job. So it was about taking, this, there we go. And this robot even spells out the names of our most important donors. So the vision, so it came about, it was a discussion between scientists and FightMND, and I really want to point out a shout out here to Dr. Ian Davis, who helped conceive this idea. And it came about through a funding partnership with the Victorian State Government, most importantly FightMND and also the Balkan Group. This is a workflow of the program. So what we did is we put a call out to volunteer MND patients to participate in our program. They became anonymised. They came in from various major clinical sites across Australia. There were two steps to this program. One was the reprogramming. The other was the motor neuron differentiations, phenotyping and drug screen. And then the idea is we would discover drug hits, which could then inform potential future clinical trials. So the crux of this program really came down to access to two robots. So the first was a reprogramming robot. So in order to reprogram hundreds of patients, we needed automation, and we were very fortunate to collaborate with Elise Peabay and Alex Hewitt and their teams at the Eye Hospital using their reprogramming robot. And then the real task came for us at the Florey. We had to establish this dedicated screening robotic platform. This is just a photo showing you our setup, and this is a team which is led by Dr. Chris By at the Florey Institute. So I'll just take you through how this platform works. We have patient iPSCs arrive. They're differentiated to motor neurons. They're then seeded into microtiter plate, high density. This robot here is an automated liquid handler. It does the media changes. It conducts the high throughput drug screening. It's then fed to an automated plate loader and then fed to a live cell imaging microscope called the CD7. And we then conduct image quantification and analysis based on that. You can see here that we keep the beanies up to date on the CD7 robot, the FightMND beanie. Okay, so the first task for us was to establish an Australian-wide ALS iPSC library. This was a Herculean effort. We had many partners and collaborations involved. In the end, I think we put a call out we had about 200 patients, and at the end we had about 150 patients and about 30 controls enroll in the program. We'll get through the QC, and this just shows you the state split. So this was a mainly Victorian initiative, but we had interest, a lot of interest from other states, which was great. This just shows you recruitment. So we started back in late 2017, and we reached our target goal of 150 patients in less than a year. This is our patient 0, Neil Danaher, receiving his skin biopsy and donating cells to our program. I just wanted to break down some of the demographics and phenotypes that we captured. The idea behind this program is we wanted to capture the immense diversity of ALS clinical, pathological, and genetic phenotypes. So what we did is, this just shows you a breakdown. We've captured about, and more importantly, we wanted to capture the sporadic ALS population, which largely gets ignored in laboratory models. So here we've captured about 90% sporadic, 10% familial. It was mainly male bias, as you'd expect. And this just shows you the number of patients captured in our program according to age of disease onset. So yes, most patients were in mid to late life, but we did capture patients with early onset ALS as well as late onset. And then this just shows you the various diagnoses. So again, we wanted to capture the whole spectrum of ALS phenotypes. And we've mainly got classical ALS. These ones have since been classified: PLS, PMA, bulbar onset, and the works. This is another way that we can depict this. So these are our patients in our program. You can see the majority of them have lower limb presentation as well as upper limb. We did capture bulbar as well as respiratory and trunk and PLS. And it was important for us not to just capture different sites of onset, but also different progression rates. And as you can see from this graph here, we've captured classic, slow progressor, fast progressor, and that was done according to ALS-FRSR progression scores. Another way that I can present this is this spaghetti plot, and this may look very familiar to what I showed you at the beginning of my seminar. So what we've done here is captured the immense heterogeneity and diversity of ALS phenotypes in our program, which we thought was important. Stage 2 involved reprogramming these cells. So as I mentioned, we had 150 patients, 30 controls. Their fibroblasts were cultured, reprogrammed, and then expanded. So this was quite a lengthy process. We did spend a lot of time on quality control and validation steps, and I'll talk about this briefly. We feel that there are a lot of iPSC lines out there, whether it's commercial or collaborative, that haven't perhaps undergone rigorous QC, and then they're distributed. So we had to ensure that our lines were rigorously validated using these 3 measures. So karyotyping, checking for pluripotency, trilineage potential. And then lastly, we had to correct some of the lines. So I mentioned that we had some familial ALS lines, and these were repaired back to wild type. This just shows you some data from our QC efforts here. So what we did is we did check for karyotypes versus the genomic DNA from the patient donors, which was all in check. And also we did check for pluripotency and trilineage potential, and that's also shown here in this heat map. So this was all good. So the third stage of this program then was optimizing our motor neuron differentiation protocol. We chose to focus on spinal motor neurons, given the vast number of protocols out there. I did want to point out that the important task here was designing or optimizing a protocol which then reflected selective motor neuron vulnerability. And we adapted one from Dew et al. 2015. It's quite a well-known protocol, and the protocol worked well in our hands. But in the early stages of this optimization, we discovered that some of our motor neurons lines simply did not die. So they did not show any neuronal vulnerability or cell vulnerability. And so over the last, I'd say 3-4 years, we've been rigorously optimizing this protocol. I did want to point out too that we don't just make motor neurons in our group. We also have the capacity to generate glial cells, so astrocytes, oligos, and microglia. And this is largely thanks to Sam Barton in our group. But coming back to motor neuron differentiation, This slide here shows you, ultimately, we ended up with a protocol with 5 different stages, of which we would conduct phenotyping in stage 5. And this would be from, I think, 28 to 60 days in vitro. This just shows you the purity of our spinal motor neurons. So they're expressing all of the correct transcription factor and neurochemical markers. So the purity of our cultures is quite higher than what is published. And we see a complete absence of glial cells contaminating our motor neuron cultures. And then this just shows you what I was mentioning. We then undertook a very rigorous deep phenotypic screen of these motor neurons from 28 days in vitro. And this just shows you we identified them unambiguously using an HB9 promoter-driven GFP reporter. And we're currently looking at various readouts. So these include looking at axonal outgrowth, cell survival, TDP43 pathology, stress management pathways, excitability. But for the purposes of this seminar, I'm just going to talk about axonal outgrowth, because we think this has enormous potential for measuring phenotypes and response to drugs. So in collaboration with John Locke from UNSW, we were able to set up a automated, an algorithm that could measure axonal outgrowth in our differentiated cells. And these are just some videos showing you the behavior of these cells over time. I'm not sure if this will play. But what you can see is these cells have a finite lifespan in culture, as you would expect, and they begin to start bleeding over time, and you see destruction of these axons. So if I just skip ahead here, I wanted to show you some data that we generated. All of this is unpublished, soon in a manuscript to be submitted. So the first thing we wanted to prove in our system is, can we detect a genuine ALS phenotype in these patient-derived cells? And we first focused on the familial ALS. So these are the monogenic lines, because we thought they will have the most robust phenotype. So what you're looking at here is we're measuring axonal length. So we've called this axonal health over time. And in these control motor neurons, you can see that they plateau at about 40 days, and then they begin to see axonal degeneration. In a patient or in patients carrying SOD1 mutations, this process is accelerated. So you can see that the rate of degeneration speeds up if you carry a SOD1 mutation. This was also true of TDP43 mutations. It was true of VCP mutations, ubiquitin-2, and also C9orf72, although the phenotype was somewhat delayed, but it did crash out at about 48 days in vitro. So we thought this is fantastic proof of principle. Others have certainly shown this before using familial ALS lines. But then the question we had is, what about sporadics? And this is the great unknown in ALS, is can you effectively model sporadic ALS in the laboratory? And the answer is yes. So we took our 100 sporadic ALS lines compared to 30 controls, and we were able to measure something called the lethal day 50. So this is the time taken to lose 50% of axons. And using this analysis, we're able to generate these plots, these survival curves. And in our system, the first thing I'll point out is that we were able to show axonal degeneration in motor neurons from sporadic ALS patients in about 30% of our sporadic ALS patient cohort. And it was significant. So here we're comparing them against healthy controls, and we can see we've got a significant reduction in survival of these cells. And to us, this was quite a remarkable finding, because we know that these patients who we have conducted whole genome sequence on, they don't carry any of those monogenic genes that are responsible for familial ALS. So we think that this axonal degenerative phenotype is being driven by, clearly, because they're derived from iPSCs, is driven by the genomic DNA, which is quite interesting when you think about the etiology of sporadic versus familial ALS. The next question we had is, after we demonstrated that we could show a phenotype in sporadic ALS lines, we then wanted to show proof of concept of our drug screening program. And we took the only approved drug for ALS in Australia, riluzole, and we treated our sporadic ALS lines with riluzole. And remarkably, we were able to show that riluzole can rescue this spontaneous axonal degeneration phenotype that we detect in sporadic ALS lines. And that's shown here on these traces. So we have an improvement in or a delay in axonal degeneration. And it's also shown here in these micrographs. So by establishing a benchmark, a critical benchmark in this drug screening platform, we're now at the point where we've started screening FDA drug libraries in reference to riluzole and other drugs. Now, I did want to mention that we're not just looking at axonal phenotypes in our system, we are looking at other outcomes. One major one of interest probably to this audience is TDP-43 pathology. I can tell you at this stage, we are not detecting robust TDP-43 pathology, at least in the limited number of lines that we have looked at. We also are looking at markers of proteostasis, so P62 ubiquitin, and also some cell death markers looking at using cell painting technology. Now, I just wanted to end by telling you that we have overlaid this program with something called a precision medicine program. And the difference with this program is once we conduct the phenotyping, we then correlate the phenotype to the genomics, transcriptomics, proteomics, metabolomics, and the clinical profiles. And then we package this up into what we call as an MND signature or barcode. And this is an attempt in order to stratify patients according to drug response, because we think drug response could be an important readout of a phenotype in this system. And I just wanted to let you know about our progress with this. We have completed our whole genome sequencing for all 150 ALS patients in our program. The RNA-seq, that should read RNA-seq, is actually in progress. And we're using proteomics and metabolomics to confirm our RNA-seq changes. So we should have a full transcriptomic package from each patient, and that will be correlated to phenotype within the next 6 to 12 months, which is very exciting. What have we found with whole genome sequencing? We have found that the familials are largely represented by, as you would expect, these major ALS gene mutations in SOD1C9, TDP-43, ubiquitinin-2, but we have also found some ALS gene mutations in our sporadic population. And interestingly enough, these are more heterogeneous. You can see matron3, ALS2, CHIMP2B, and NEC1 amongst that pool. Okay, so I just wanted to finish by mentioning, yes, there are limitations to iPSCs. I think it's very important that the QC is performed at a very high standard, and that there is rigorous phenotyping of these lines before they're considered for distribution to other groups and labs. One of the areas that we're working on in the lab is really looking at how can we improve or refine iPSC models. And this is really about building three-dimensional models of ALS. And I mentioned to you before that there are other cell types critically involved in ALS pathogenesis. And this is a shout out to work by Dr. Sam Barton in the group, and also Lee John Lowe, who's a PhD student. And the two of them have been working on these protocols, generating these human spinal cord organoids, as shown here. And I'm just going to take you through some quite striking images. So these are three-dimensional spinal organoid constructs derived from ALS patient iPSCs. And what we can see is that they contain, firstly, motor neurons. They contain interneurons, as shown by these markers, calbindin. We're just going to take a slice through one of these spinal cord organoids here. You can see really striking examples of alpha motor neurons in these organoids. Importantly, they do contain quite abundant astrocytes, as shown here with these two markers, vimentin and GFAP. They do contain myelinating glia, at least oligodendrocytes, as shown by the expression of these critical markers, especially MBP as a marker of mature myelinating oligodendrocytes. And I'd just like to show you these images of myelinating human spinal cord organoids with time, and you can see that these myelination profiles become quite extensive in about 12 weeks in culture, and then you end up with striking images like this. We have stained neurofilaments with MBP, so we've got nice myelinated axons. And lastly, you're able to add microglia with a bit of innovation into these organoids, and you end up with these really quite striking images. So I'm just going to end by showing some really impressive data that Li Jun has generated. So Li Jun is working with C9 orphanib2 expanded ALS patient organoids, and she's taken C9 lines as well as isogenic controls, and she's creating these spinal cord organoids. And at this stage, her preliminary data suggests an ALS phenotype, well, actually two phenotypes. So if we look here, there appears to be a shortening of axonal length in the motor neurons in these spinal cord organoids. And you can see that the isogenic control has longer neurites. And interestingly enough, there is a hint that these organoids are exhibiting hyperexcitable networks, and you can see that summarized here in this graph. We've got a hyperexcitable train of potentials versus the isogenic control. So this is a really exciting, I think, evolution of the 2D iPSC model. So I'm going to wrap up here, and just to conclude my talk, so today I've presented an update on the ID MND Initiative. And this is really a large-scale population-wide library of ALS patient iPSCs across Australia. And the idea is that we wanted to encompass that enormous clinical, pathological and genetic heterogeneity that we see in patients for the purposes of capturing all of this in disease modelling, to undertake some really deep disease profiling, to understand differences in these patients, what confers their phenotypes, and then how we can stratify them in a more intelligent way. And also it provides us with a vehicle for drug screening, particularly personalized drug screening. I would point out here that we were able to show that even motor neurons differentiated from sporadic ALS patients, or at least a proportion of them, do reproduce pathological phenotypes in this system. And what we've detected here is evidence for spontaneous axonal degeneration, which I think is an important advance. Also, we're able to show that these motor neurons generated from these patients can reproduce the efficacy of riluzole, which is the only approved drug for ALS. And this really sets the scene for our ongoing high throughput drug screening activities. And lastly, I think that these cells, these iPSCs, will provide, and they continue to provide, a very relevant drug screening tool for looking at precision medicine approaches through the use of drug repurposing. So I just wanted to end by acknowledging the cast of people behind this, but firstly, to those MND patients and their families and carers who came aboard in this project and volunteered and donated samples. This project wouldn't exist without that type of investment from the public and the community. And then I'd really like to thank Dr. Chris Bai and his team, so Catherine Lim, Elizabeth Kwan and Duncan Crombie, who have really superbly, so Chris manages this facility, and all of them have really played a critical role in getting the ID MND initiative to where it is today. And thanks also to Sam and Joel and Li Jun. And also thanks to our clinical partners in this program. So these are the clinical sites across Australia. So Susan Mathers, Paul Torman and Marilee Needham. And of course, thanks to Elise Peabay and Alex Hewitt and their teams at CIRA for the reprogramming. And then we've got help from Naomi Ray and her team with the genetics, unpacking that genetics. Most importantly, thank you to our important funders, FightMND, Victorian State Government and the Balkan Group. Thank you all, and happy to take questions.
Thanks, Brad. An enormous amount of work across a number of really exciting projects. I'd like to open it up for questions, and if you are asking a question, if you wouldn't mind introducing yourself, just your name and your affiliation. So firstly, we've got Lizanne. I feel like we've just lost her. Lizanne, you should be able to ask your question.
Oh, great. Thanks for unmuting me. I was trapped. Hi, Brad. I'm Lizanne Uy from the University of Wollongong, and that was a lovely presentation, so thank you for that. I think the axonal degeneration phenotype that you've described is a really great finding. So I understood from your presentation that you were saying that you didn't actually see that phenotype until you modified the DO protocol to fit your needs.
Yes.
So So I guess that's a challenge for the field in general, because there would be many people using protocols that are potentially not fit for purpose. So yeah, I guess that's potentially a major issue for the field.
It is, and it was challenging the first 3 years that we got into this. We just couldn't reproduce phenotypes in these cells.
Yeah. So is there anything that you're able to share about those changes to the protocol that allowed you to kind of unmask that phenotype? And then how variable, so I think one of the huge strengths of your project is the massive number of lines that you've used. So how many lines do you think that you have to incorporate into the drug screening component to be able to identify a genuine drug target?
Yeah, great questions, Lizanne. So I would say all should be unveiled in the script when it's published, or it could even be a preprint. But yeah, it did take many years of dedicated optimization. And I would hint at it that perhaps minimal media is best. So withdrawing growth factors and antioxidants and things that you think are critical, for preservation of motor neurons may actually help increase disease penetrance in the simplistic iPSC model. The second question is yes. What we've done now is I think we've generated a primary panel for our drug screen, where we take perhaps 10 familials and 10 sporadics. We're very interested in that 30% of sporadics that were disease penetrant versus the 70% that were not. And we're hoping that the RNA-seq or even the ePhys will, reveal the secrets or provide hints behind that. But yeah, that's probably a sizable number that we'll be using going forward. And then we will use the rest of our patient cohort as validation.
Yeah, understood. Thanks very much.
Thank you.
Thanks, Suzanne. Can I ask you, Brad, about the 30%? And I know we've touched on this previously, but around the 30% of patients that did demonstrate that phenotype, around what you're doing to look at the clinical data around those patients and whether or not there's something about them that makes them more susceptible.
Yeah, sure. So we weren't surprised by that outcome. We never expected, 100% of our lines would show a phenotype. I think it could relate to the phenotype of these patients, and that's something that we need to correlate. So is it something unusual about the disease sort of onset rate of progression? Intuitively, you would think it would be that, but we suspect it's perhaps something more subtle. And as I stressed, we're only looking at the spinal motor neuron component of ALS. We didn't model the cortical motor neuron component or the upper motor neuron component. So perhaps that could account for the missing phenotype or the lack of phenotype. The other thing I didn't really talk about in the seminar was I may have portrayed this program as straightforward, but those that work in this field with stem cells know that it is plagued by technical and biological heterogeneity and variation. And I think that 70% could be accounted for through those factors. So I think we may need to increase the complexity of our system. So we need to maybe perform co-cultures with muscle or astrocytes, or just jump in the deep end and create these three-dimensional spinal cord organoids in order to tease out a phenotype, I think, from that remaining 70%. We're very interested to know what the molecular signatures are that differentiate that 30% from the 70% in our system.
Thanks, Brad. Got a question from Naomi Ray.
Hi, Brad. Thank
Hi.
you for a really nice talk. I'm just a bit out there, but what do you think about the village in the dish type technology? Is that another just too far in the technical complexity? I mean,
yeah, so that term got thrown around, I think, by Kevin Egan at a conference a few years ago. We tried to pool our motor neurons in the dish and then try and barcode them in a way using the genetics and the transcriptomics, but it just failed. And I haven't seen anything published about, village in a dish. I think that would certainly help with uniformity of the phenotypes, and that's what I'm getting at. This biological technical variation that's plaguing this field. We tried, and we haven't seen anything published, I think is the answer.
I think Kevin Egan has published, and I think Joseph Powell has been looking at it, but not with neurons, of course.
Yeah, do you know how many lines?
I find it an interesting concept.
Yeah.
Because of course you could increase your N in a big way, and
Yes.
have different people in different villages, and so from a conceptually it's very interesting, but technically very difficult.
Yeah, we're certainly open to building villages.
Yeah, thanks. It's really great work, really great platform.
Thank you, Naomi.
Thanks, Naomi. Brad, I might ask a question. Obviously, you've got a paper in preparation for submission. So what's the next steps for the platform, and what other programs do you have in mind?
Yeah, so we certainly want to step up our organoid program and increase the complexity of organoids. So that, I mean, the organoid field moves day by day, and they're already talking about assembloids. So these are three-dimensional constructs where you can connect a cortical organoid with a spinal cord organoid with a muscle. So it's built, and generate neuromuscular organoids. That's one area we'd like to go. I think that, the next critical step for us is now that we've got our rigorously, robustly reproducible phenotype lines, we know which ones to use in the primary panel for our drug screen. Now we move on to true high throughput drug screening. I will point out that we did screen a couple of other drugs that were in clinical trial for ALS, and so we have some negative controls, if you like. These are drugs that spectacularly failed in ALS clinical trial, and we couldn't generate any signal in our system. So that's concordant with what's happened in the clinic. So I think we'll have focused drug screens where we take drugs that have already been assessed for ALS, and we have new drug screens where we repurpose FDA-approved therapeutics. We'll also open up to new drug libraries as well. So huge scope there.
Thanks, Brad. Any more questions from the floor? Naomi, do you have another question, or is your hand still up? I think her hand.
Sorry, my hand was still up. Yeah.
That's okay. Okay, well, we might leave it there.
I'll ask a question of Brad. Brad, in terms of phenotyping, axonal degeneration seems very robust there. Were you surprised by the lack of TDP43 accumulation in your cell lines? And have you looked at any other proteins or evidence of proteinopathy?
Yeah, so not surprised. I remain agnostic about the relevance of TDP43 pathology in ALS. Interestingly enough, if you look at the literature, no one has really reported any robust TDP43 pathology without the application of a stressor, like a chemical. I think there is increasing evidence that TDP43 can mediate damage even when it's nuclear resident. So I think the next step for us is we need to look at other sensitive markers such as TDP43 phosphorylation, just not where it's located. And we're looking at proteostasis markers and biosensors at the moment. So we think the other thing to keep in mind too is that these cells, they do have a phenotype, but they're also what I would consider there's still in the infancy, I think, of ALS pathophysiology. And I think if we can start to maybe exclude or rule out some of these reported pathologies in ALS, then that could also be informative for the field. So that's where we're at with that.
Thank you.
Question from Garth Nicholson. I'll just allow you to, should be able to turn on your camera and mic now, Garth. Yeah, you just need to unmute yourself, Garth.
Perhaps Garth can type his question. I'd love to hear it. I suspect it could be a genetics question.
If you're having trouble unmuting yourself, you can
Okay.
type it in the chat. We've got another question from Mary Louise Rogers. Mary Louise, you should be able to unmute yourself.
Okay. Just Brad, I was just going to ask about electrical properties. So are you saying that it's more like in the organoids? Is that where you get the electrical properties of these cells?
Yeah, we have patched them in 2D, and they are excitable. I would say that you get more mature networks in the organoids, as I showed you in that data, and perhaps that's a better way of teasing out excitability phenotypes.
So when you give riluzole and that, you should, do you think, or things that are going to affect the excitability, you'd be able to get a readout sort of thing?
Yeah, certainly. So that what's interesting about our simplistic 2D system is the fact that riluzole works at all. Does tell you that it's working cell autonomously without astrocytes in the system. So it is blocking sodium channels or antagonizing glutamate receptors. It almost gives you some insights into the potential mechanism of action of riluzole, which has been debated for decades.
Interesting.
Yeah, I agree. Thanks.
Thank you very much.
Thanks, Mary Louise. Garth, I can still see you've got your hand up. If you are having trouble unmuting yourself, just give it another minute. Oh, here we go.
Can you read that one out, Beck, or it disappeared on me?
Got a message saying only select people could unmute. Brad, can you reduce heterogeneity of causes by using a N of 1 approach?
Right, so the old N of 1. I guess that the whole point of this initiative was to go large scale population wide, in the hope that we could then segregate out subgroups of patients that were drug responders, or subgroups of patients that have the same excitability phenotypes or transcriptomic signatures. So I would argue at this stage we're staying with population wide.
We have another question in the chat from Serene Kim, who is a postgraduate student studying ALS at the University of Auckland. And they've asked, have you observed any differences in glial cells between ALS and control in your stem cell and organoid models?
Yeah, thanks for tuning in. Yes, we have. So Dr. Sam Barton has been looking at the behaviour of astrocytes and oligodendrocytes derived from ALS patients. And I believe that she has some interesting phenotypes. So yes, there are altered behaviours of glial cells. So our focus with this initiative has been purely on the lower motor neuron, and we're quite keen to branch out to upper motor neuron, muscular and also glial cells. Thank you for your question.
Questions? Okay, we might wrap it up there for today. Please join me in thanking Brad for a fantastic talk. If you have any questions for Brad that you'd like to follow up, we're happy to share his contact details so you can reach out. And stay tuned, we're going to have one more seminar this year in November, and we have a fantastic speaker lined up for that. So keep your eye out on your inbox for announcement of registrations for that in the coming weeks. So thank you again, Brad, for joining us, and for everyone for tuning in today, and we'll see you next time.
Thanks all.